This is the second part of a two-part video series where we looked at which companies may actually win the race to commercial success for the treatments of beta-thalassemia and sickle cell disease. If you have not seen part 1 of this video, I'll leave a link here as well as in the description below. Although I have a PhD in biomedical engineering and over 15 years of working experience in the healthcare industry, I do not intend to give you investment advice. Please do your own research before making any investments. You are watching Health, Wealth. If you're new to this channel, welcome. If you're not, nice to have you back. Before we continue with part 2 of this video series, I'd like to address a viewer's question. In fact, I'd like to announce that I intend to address viewer questions more regularly as part of my video series. I realize not everybody may read and follow up on the various comments posted to my videos, so I think that more viewers will benefit from the relevant and very important questions that many of my viewers actually pose in the comments. And I believe that the answers will reach a wider audience when I include these replies in future videos where contextually applicable. I hope you will enjoy these additions to my videos, and I encourage you, if you have questions, don't hesitate to post them. However, if you do and you are not yet subscribed to my channel, may I please ask you to do so first. Mathilde is asking about the global prevalence for beta-thalassemia and sickle cell disease. Bristol-Myers Squibb listed the answer on their website in an article published in 2018. As always, you'll find a link to this article in the description below. And in the article, they mention that each year, more than 60,000 infants are born with the disease. For sickle cell disease, the Journal of Global Health holds the answer to the question of the global prevalence. You'll also find a link to this article in the description below. This metadata analysis lists the prevalence of sickle cell disease as 112 per 100,000 live births. However, there is a large geographic difference. In Africa, it can be as high as 1,125 cases per 100,000 live births, whereas in Europe it is only 43 cases per 100,000 live births. So Mathilde, thank you very much for raising this question, and this provides a nice segue to the continuation of this video series with part 2, where we now focus on orphan drugs. So let's now consider what orphan drug status in this context actually means. In Europe, an owner of an orphan drug enjoys 10 years of market exclusivity. And in my previous video, I had suggested that perhaps CRISPR Therapeutics may be able to commercialize their treatment for beta-thalassemia, at least in Europe, sometime after January 2023, because that would have been 10 years after the orphan drug designation was initially awarded to Bluebird Bio. This assumption, however, I have to correct. As I did more research, I came to realize that the market exclusivity is actually granted from the time that the marketing authorization for that particular medicine is granted. And in the case of Zynteglo, the marketing authorization in the EU was granted in June of 2019. So we may then need to consider under which circumstances the 10-year exclusivity period granted to an orphan drug may actually be shorter. And while I have not yet been able to find this information directly on the EMA website, I would like to thank my viewer Frank Lee for pointing out that this information is contained in the Bluebird Bio annual report. I leave a link to it in the description below. So, if a second applicant can establish that their product, although being similar, is in fact safer or more effective and otherwise clinically superior, may actually obtain orphan drug status and the market authorization, even though a previous product should in theory still be under market exclusivity. And the original beneficiary of the market exclusivity for an orphan drug may see a competitor coming into the market in this way. Under the US FDA regulations, the orphan drug status exclusivity period is 7 years instead of 10. Likewise, when a product with clinical superiority is presented, the FDA may nevertheless approve the product, even though the earlier product may still enjoy orphan drug exclusivity. So, now that we understand the rules of the race, how do these competing companies stack up in the race to a cure for beta-thalassemia and sickle cell disease? They all focus on ex vivo cell editing in order to induce the formation of fetal hemoglobin as a treatment for the diseases. Apart from different delivery methods for the ex vivo editing and delivery of the CRISPR-Cas component to induce the desired gene edits, there might be only few features that could distinguish and separate clinical superiority from one gene edit or one company over the other. Induction of fetal hemoglobin allows alleviating the disease symptoms. However, strictly speaking, it is not actually a cure. Fetal hemoglobin can certainly address the most severe forms of the disease and prevent the frequent blood transfusions. However, I wonder if there's a downside to inducing fetal hemoglobin, and in a previous video, I talked about an assumption that due to the higher oxygen affinity of fetal hemoglobin, patients treated in this way might find themselves more easily out of breath, a condition that is called medically hypoxia. I thank another of my viewers for challenging me on this particular point and raising the question about elevated levels of fetal hemoglobin, exercise intolerance, and hypoxia. So, based on this, I did additional research in the peer-reviewed medical literature and came across this. In this journal article of critical care is an interesting quote I will highlight next, and as always you'll find the link to this journal article in the description in the video below. So, interestingly, in patients with sickle cell disease, fetal hemoglobin production, or HbF as abbreviated in this article, continues to be produced into adult life and optimizes arterial oxygen content. In fact, fetal hemoglobin production is of such importance in the sickle cell patient population that low levels of it serve as an independent predictor of early mortality. The next few lines of the article address my viewers' question, or rather confirm their statement. There is a genetic disorder whereby those people afflicted by the disorder only produce the fetal form of hemoglobin, and it appears that this is a benign condition not affecting these patients at all. In fact, we may not even consider them patients at all. I thank my viewer for raising this point and challenging me it, and with this I'd like to correct the statement I have made in the previous video. With regards to fetal hemoglobin production under normal condition, the following happens. The transition from fetal hemoglobin to adult hemoglobin happens soon after birth, and it is achieved through the silencing of the gene expression responsible for fetal hemoglobin. As far as I understand, all of the CRISPR-Cas gene editing techniques to activate fetal hemoglobin do so through the non-homologous end joining, which induces so-called indels, that is, insertion or deletions in the genetic code at the desired target site. And it is this indel formation which you could liken to the destruction of a house, so to speak, which then actually breaks the silencing of the gene expression for fetal hemoglobin, and therefore fetal hemoglobin is activated again. You can then think of this indel formation, which breaks the silencing of expression for fetal hemoglobin, as perhaps somewhat of a random process. But as long as the silencing part of the gene expression is successfully broken in any way, the end result is achieved. And just like there are multiple different ways you can destroy a house, there are multiple different ways of how an indel can finally break the silencing of the fetal hemoglobin expression. So, from a clinical point of view, clinical superiority may manifest in the efficiency of the edit, in the efficiency of how the CRISPR-Cas component for this type of edit can be delivered ex vivo to the appropriate cells. However, in my opinion, it will be very difficult to argue that one type of indel formation would be superior to the indel formation of a different kind, because in both cases, the end result is that the silencing of the fetal hemoglobin expression is achieved. And this is precisely why I said earlier that we should not discard Beam Therapeutics, who seem to be very late to the race. While the Beam 101 study follows suit to all the other companies in the attempt to induce fetal hemoglobin, the true unique nature of Beam 102 is in fact a genetic edit that will correct for the gene mutation that is causing sickle cell disease. And while this program does not address beta-thalassemia, I would argue that a true correction of the gene mutation is in fact what constitutes a true treatment, and therefore, in my opinion, should provide clinical superiority over all the other treatments who manage to address the disease through the induction of fetal hemoglobin. And this is ultimately where Beam could come to shine. Granted, the road to commercial success for Beam with their 102 program may still be many years down the road, in which case the other companies, or whoever brings treatment for sickle cell disease and or beta thalassemia to market, could still possibly enjoy many years of orphan drug status exclusivity before a treatment provided by Beam Therapeutics could pose a real threat. So, what does this mean for all the other companies working on a treatment modality through inducing fetal hemoglobin? FDA fast track status, comparable to the European priority medicine or PRIME designation, could lead to a regulatory pathway that is faster than under normal circumstances. With superb clinical trial data, it could be possible that a market authorization could be granted even though phase 3 clinical trials are not yet fully complete. So overall, it could still look very much like a neck-to-neck race, and with orphan drug designation, it would definitely seem that the company who manages to get the market authorization first probably will be the company that enjoys market exclusivity for either 7 years in the US or 10 years in Europe until another company can come along and prove better clinical efficacy or safety. So, before wrapping up this video, let's take a look at what clinicaltrials.gov can tell us how far along in the race to a cure each one of the companies are. Let's go by disease indication, and we'll start with beta-thalassemia. Unfortunately, not all studies are listed on clinicaltrials.gov, and I've only been able to find the information for 3 different companies. Let's start with Bluebird Bio. We see that their phase 3 clinical trial started in June of 2017 and is planned to be concluded in June of 2022. We of course also know that the treatment is already commercially available in Europe. We then find for Sangamo Therapeutics that their phase 1 and 2 clinical study started in March of 2018 and is slated to be completed by March 2023. We then have CRISPR Therapeutics with their CTX001 study, also phase 1 and 2, which started in September 2018 and is planned to be completed in May 2022. So, in summary, apart from the fact that Bluebird Bio complete a phase 3 study in the middle of next year, and CRISPR Therapeutics conclude their phase 1-2 clinical study, it appears that Sangamo is still 1 year further behind, with their completion by March 2023. So let's then switch to the trials for sickle cell disease. On clinicaltrials.gov, we find the EDITAS study phase 1 and 2, which started in April of 2021 to be completed by August 2025. Next up, the Intellia or Novartis study, also phase 1 and 2, which started in August of 2020 and to be completed by December of 2023. Next up again, we have CRISPR Therapeutics, and their phase 1 and 2 clinical study started in November of 2018, also to be completed in May of 2022. And unfortunately, I did not find any listings for clinical studies from Sangamo Therapeutics or their partner Sanofi. So, if we summarize here, for sickle cell disease, it appears that CRISPR Therapeutics is actually ahead of the game, with their phase 1 and 2 study to be completed by May 2022, whereas the others basically look at December 2023 or even August 2025. If this video provided any valuable information to you and you feel I deserved it, I would definitely appreciate you subscribing to my channel and liking the video. Thanks in advance.